anybody here today that was not here on Monday? Okay, so I've got two. So we are going to do some group work on next Monday. And what that's going to entail is you have to go out into the literature and you need to find an article that pertains to a reaction of glycolysis, of which I'm going to assign you. So you will be reaction number 3, and Mungid, you'll be reaction number 4. So I assigned the groups last week, so each person has to find a paper, and so this was what the email, how many of you got the email and read said email? Okay, so some of you. So I sent an email clarifying that. So the numbers 1 through 10 correspond to the reactions of glycolysis. So if you got number 1, your reaction number 1. And again, what you need to do is you need to go out and find a peer-reviewed paper that will help you fill in the blanks on these worksheets. And so you'll do that together in groups on Monday, and then you guys will turn that information, well, actually I'll have you hang on to that. You'll turn in your papers at the end of that group work, and then you will, everybody in class needs to download this handout. And it's not only on Blackboard, but I also posted it on my website. So has everybody been able to find it, get it, that's looked for it so far? Any issues with getting the file? Okay. So make sure that you come to class with this starting next week on Monday. We're going to use it over the course of the next several weeks as we get through some of these pathways. And I will be adding more worksheets like this. This is just the first packet. So if you are unclear on any of this, let me know. And for the people that are joining us new today, make sure that you talk to your classmates. Find somebody in class to talk to, or come see me and I can explain it in more detail. So, everybody good with what we're doing on Monday? And again, I'll probably have you guys work on that for about 20 minutes or so. Usually that's plenty of time for each of you to do your assigned reaction. And then you don't have to worry about what the other groups are doing because everybody's going to share that information once we get to that point where we're ready to talk about the individual reactions. Okay. The only other thing that I have announcement-wise today is that the Chapter 14 homework is now open on saplings, so that's going to stay open until next Friday, and so it'll be due by 9 a.m. So by the time you guys come to class next Friday, the 6th of February, make sure that you get in and do that homework. Last semester, I think I had a handful of people that for some reason or another did not complete 80% of the homework, and they got no credit for homework. So don't let that be one of you. Just make sure that you're going in and consistently working the homework. It's good practice. The questions I don't think are particularly difficult, but it's really just the process of getting in and thinking about some of these things. If you want some more challenging questions, I recommend that you go to the back of each chapter. Those questions are a little bit more essay type questions, and because of the format of sapling, you can't really do essay questions on sapling. It's more multiple choice format, or draw me a structure, or something like that, or fill in the blank. So the questions I think are fairly straightforward. Again, if you really want some more challenging type questions, I would always recommend that you go to the back of the chapter. And I believe half of the answers at least are at the back of the book. So any questions about that? So always do your homework. Don't let those 50 freebie points slip away, because really they're the easiest points to get in this class. Okay, so what I want to do today is I want to start talking about some of the more general characteristics of metabolism, just so that when we start looking at the individual pathways, you're familiar with some of the regulatory aspects that we're going to be seeing. And again, you're going to become very familiar with, oh, okay, that particular reaction is regulated by allosteric effectors, which can either be positive or negative effectors, or it can be a covalent modification, or it can be hormonal in nature. There's a lot of different ways that these reactions are regulated, and so really looking in depth and figuring out what the mechanism of regulation is. And again, it can be multiple types. If it's a really important reaction, you're going to find very heavy regulation. And then part of what we're going to do is really think about why. have to regulate that particular step so heavily compared to other steps that are freely reversible. So there's a lot of things that go on, and again, this is just really to give us the initial flavor of how everything's going to work, and you'll start seeing patterns. So you'll start seeing these things over and over again, and I want you to become very familiar with them. So first thing I want to do is just start with some very basic definitions. Metabolism itself is, in the most simplest terms, the sum of all reactions in a living organism. And as such, probably makes a lot of sense that it is required for life. Basically, all of these reactions put things into a time frame that is useful for a living organism. Whereas if you think about it, if you have an uncatalyzed process, like let's just say digestion of a meal, if you don't have enzymes to help you break down food into the nutrients that you need, you're probably going to starve to death before you get out the energy that you need. So the enzymes are there to help us, and the metabolic pathways are there to help us to do things on a timeline that helps us live. Okay, because we can't wait for things, and the stuff's going to rot. If there were no enzymes, the food would rot before anything good came out of it. So that would not be a good thing. So we can really break down the types of reactions that we have into two main categories, anabolism and catabolism. So anabolism is the biosynthesis of biomolecules from smaller precursors. So we start with little things and we make big things out of it. The catch is, it's uphill energetically. Forming bonds has an energy cost. So we're going to see, for those pathways that are anabolic in nature, we're going to see ATP equivalents in to make that happen. Okay? So you're going to see some sort of an energy input. So catabolism or catabolic pathways is the breakdown of molecules into smaller components. So taking those big things and breaking them down into smaller parts. And in that process, we can generate energy. So the energy contained in the chemical bonds that we're breaking and rearranging gives us energy output. But then we can do things like fuel anabolic processes. Okay? Now there's one other term I want to give you at this point. It pertains more to pathways. And we'll see this, not chapter 15, but in, I believe, the very next chapter after that. So I'm going to quiz you guys about something from last semester. If I say something is amphibolic, what would you think it means? It's both. So that means that the pathway is going to be both anabolic and catabolic in nature. So it's a dual purpose pathway. Some of the reactions lend themselves to being catabolic, and some reactions lend themselves to being more anabolic. So the very first pathway that we're going to see that meets this criteria is the citric acid cycle. So it has elements that are both anabolic and catabolic in nature. So we'll see that very soon. So as I mentioned on Monday, we're going to be looking not only at individual reactions and talking about them in great depth in terms of what's happening, why it's happening, if it's regulated, how it's regulated, and why, but we also have to think about the bigger picture, which is how does this particular reaction fit into the scheme of things in a multi-step pathway. So again, glycolysis has 10 steps, so each of those reactions plays an important role in the pathway's function as a whole. And so we have to always think about what's going on with respect to the pathways themselves. And then even beyond the pathways, how does that one pathway fit in with all of the other pathways? So that lovely picture that I still need to scan and post for you guys, which is on an 8.5 by 11 sheet of paper, everything you need to know for this class in one piece of paper. So I'll post that. I just have not been home in the evening lately. So characteristics of the pathways, the catabolic pathways, what we tend to get is diverse molecules in to the pathways. Okay, so things that are diverse are going to come in, but we have very few common intermediates. So there's not a lot of overlap. Another part to this that we're going to talk about in much greater detail is that in general, when we're looking at a catabolic pathway, we're looking at oxidizing carbon. And by oxidizing our carbon up, we are going to be able to release some energy. So that's going to be a very common characteristic that we're going to see. With anabolic pathways, there's really only a few things that we start with in terms of our building blocks. So our starting materials. But we get a great diversity of our products. So you'll be amazed what precursors we can use to build a whole array of different kinds of products. And something that doesn't really fit well into any of these categories, but really bears mentioning at this point, is this idea of compartmentation. And what I mean by compartmentation is basically if we have two pathways that oppose one another, and the example that I gave in class on Monday was glycolysis and gluconeogenesis, those don't occur in the same part of the cell or even the same part of the body. Okay, they have to occur in a different place because otherwise if they occur in exactly the same place, you run the risk of having what's known as a futile cycle, which is where you're just going in a circle and you're wasting resources and energy to do so. So by separating them or compartmentalizing them, there's greater control that the organism has over which pathway is activated and when. Okay, so which pathway is operational at a given time or over another one. So compartmentation of pathways is really important. Allows different processes to occur in alternate locations, and it prevents futile cycles, which is a bad thing. So one other aspect that we need to talk about is how are we really going to do our bookkeeping when it comes to looking at these pathways? And boy, that's really not a good board. And it's not going to be a good board. All right, we'll skip that one. And we're not talking about reproduction. Okay. So with respect to our bookkeeping, we are going to use thermodynamics. In particular, we are going to be looking at Gibbs free energy as kind of our measure of what's happening within a given reaction and also overall with respect to a pathway. So that's going to be our way of bookkeeping. So I want to talk about the different forms of delta G that we have, and also make you aware of one that you might not have seen before, one or two that you might not have seen before. One is used in your book a lot. The other one is not used in your book, but my reason for giving that to you is because you may see it someplace else. So people use them interchangeably. I want you to be aware that they mean the same thing. It's just a different way of stating that. So delta G itself is actual free energy change. So it takes into account actual conditions, concentration, etc. Okay, so if you have a difference from what the standard free energy change is, you would want to use the standard free energy change plus the conditions that are different to help you solve then for the actual free energy change. One you remember probably quite well from both Gen Chem and at this point probably P-Chem as well, because I think most of you have probably done P-Chem by now, is our standard free energy change, 1 atmosphere, 25 degrees C, 1 molar concentrations. Do you think those conditions apply very well to a biological system? No. Okay, 1 molar concentrations, we're going to be talking like millimolar, micromolar concentrations. So it doesn't work very well, and I'll get to another definition for a biochemical standard free energy change in a second, and really the big difference is that the biochemical standard free energy change takes into account pH, because pH is incredibly important in a biological system. We spent a lot of time last semester talking about how systems are always buffered. We have to have the right pH in order for these enzymes to work. And so the standard free energy change doesn't really work very well for our circumstances because the pH is all wrong. We also have delta G prime. What I will tell you, this is not in your book, but never fear. Delta G and delta G prime mean the same thing. So again, it's just a syntax issue. Some people will use delta G prime because they want to alert you to the fact that you're talking about a biological system. So whenever you see that prime, you're talking about something in a biochemical system. So this is the biochemical actual free energy change. So actual conditions, concentrations, but likely pH equals 7.0. Okay, so you're going to see the pH taken into account. But the pH could also be something different from this, depending on where that enzyme is functioning. So pH 7 is just close, but not every body compartment or even every cell organelle is going to be at pH 7. But this particular delta G allows you to correct for that. So likely pH 7, but not always. One your book does use is the delta G 0 prime, which is our biochemical standard free energy change. So 1 atmosphere, 25 degrees C, 1 molar concentrations except for our H+. That one by definition is pH equals 7. Okay, so 1 times 10 to the minus 7th molar under those circumstances. So if we were looking at an equilibrium, A plus B gives us C plus D, we can start to incorporate some of these delta G values into this and help us solve for what we want to know. So if we need to know what our actual conditions are, we can look up our standard free energy change, our biochemical standard free energy change, plug in our concentrations. If we have a different temperature, we can take that into account, and we can solve for our delta G. And again, this can also be delta G prime. Okay, so just simply using that equation. If we are at equilibrium, however, we can simplify it further. So delta G equals 0, and again, delta G prime then equals 0, so then we simplify that particular equation. To minus RT ln KEQ. Okay, so these are some things. I know you have some homework in this area. This is really standard stuff. You've done it before, probably a lot. So there will be some additional problems to work in the homework that relate to free energy change. Does anybody have any questions up to this point? While I'm erasing this board, which doesn't erase very well. Good arm workout though. Or maybe it's just the eraser stinks. Any questions? No? Okay. Really straightforward stuff. Another thing that we have to think about is metabolic flux. And again, I sort of talked a little bit about this on Monday, the fact that if we perturb something or we regulate a step of our pathway, that's going to have an impact both upstream from that particular step in the pathway and downstream. So when we talk about how things are moving through the pathways, that's called metabolic flux. So how are the molecules moving through the pathways? And again, if we start regulating something, that has both upstream and downstream impacts on that pathway and even other pathways that it feeds into. So one of the things that we need to talk about are these reversible versus irreversible steps in our pathways. So some of you, if you've looked through that packet that you downloaded, have probably already noticed the not so subtle differences between some of the reactions. In some instances, I used the equilibrium arrows, and in other instances, I simply used the arrow going from reactants to products. That is your first big hint as to whether or not something is a reversible or irreversible step, and also it's your second big hint as to where you are likely to see regulation. So do you guys think you're going to see a lot of regulation on freely reversible steps? Where are we going to see the regulation? On those irreversible steps. Okay, those steps that go only to product. So when we look at our reversible, our delta G is going to be very close to 0. It's probably not going to be exactly 0, but it will be a very, very small number. So it might be a small positive number or a small negative number, but it's going to be small. So small, positive, or negative number. And that means that if we alter the concentrations of either our reactants or our products, we can alter the direction that that reaction is going to go. Okay, if we increase the concentrations of products, we're going to force it to go back towards reactants. If we do the opposite, we're going to force it towards products. So controlled by the concentrations of substrate and product. So if we alter those, we can alter the direction of that reaction. Irreversible is very different. delta G is usually way, way less than 0. So it is a large negative number. Okay? So it's going to only go in one direction. You cannot reverse this reaction. So unaffected by changes in the concentrations. So if we come back and we look in greater depth, at those irreversible steps, we're going to be able to get more of an insight into what's going on in the pathway itself. So the first thing to know is that irreversible steps lead to the pathways being irreversible as a whole. Okay, glycolysis only goes from glucose down to pyruvate. If it does something different than that, we are not talking about glycolysis anymore. Okay, for those 10 enzymatically catalyzed steps, because we're going to have some irreversible steps in there, it's going to go from glucose to pyruvate. Okay, so that those irreversible steps determine the direction of the pathway as a whole. So it can't go backwards. Glycolysis doesn't go from pyruvate to glucose. We're talking about a completely different animal. When we're talking about that, we're talking about gluconeogenesis. Different pathway. Some of the enzymes are the same, but not all of them. And that's the key difference. So if we have two pathways that oppose each other, yes, those freely reversible enzymes with delta G's close to 0, we can use them in both pathways. That's fine. But number one, they're compartmentalized. The pathways are in different locations in the cell or in the organism itself. And number two, we cannot have exactly the same enzymes for both pathways. There have to be different enzymes. And those different enzymes are these guys. The ones that catalyze those irreversible steps, those are always going to be different from pathway to pathway. So these steps are going to be unique to the pathway where you find them. Okay, they're not going to be used in multiple pathways. So irreversible steps, that makes the pathway irreversible as well. And then the other thing to look for is that one step, sometimes it's more, but typically one step, early in pathway, that is irreversible. So look at glycolysis and you'll see what I'm talking about. That is not only unique, but it's also highly exergonic, meaning we have a large negative value on our delta G for that step. When we have that, that becomes known as our first committed step. Meaning if the substrate for that particular step, that committed step, makes it through that reaction, it has to finish the pathway. There's no turning back at that point. Okay, so that first committed step, you're going to see the most regulation with that. And you're going to be a little bit surprised to find out that it's not necessarily the one that you think it is in glycolysis. Because there's two regulated steps very early in the pathway. One of them is considered the first committed step, the other one is not. So we'll see that, but it's probably not the one that you think it's going to be. So that's a really important thing, and it's a clue for you guys to look for, is those irreversible steps, because that's where you're going to typically find the regulation. Okay. So the final thing that I also mentioned is that you can't have opposing pathways using exactly the same enzymes. It's okay for them to share some enzymes in common, but you're going to have some unique enzymes for each step. Because again, if you have exactly the same enzymes catalyzing two different pathways, you have a futile cycle, which is a bad, bad, bad thing. We don't want that. So if we have an irreversible step, we can pretty much guess that it's going to be regulated. But what are the different ways that it can be regulated? Some of this is stuff that we talked about last semester in Chem160A, and it pertains here as well. So when we were talking about enzymes, we talked about different ways of regulating enzyme activity. A lot of those things hold true here. It's just now we're going to add some refinement to that and add some further information that wasn't really relevant for a general discussion of enzymes, but definitely is relevant when we're starting to talk about metabolism. So that metabolic flux control so mediated through the rate determining step of our pathway, which is again our irreversible or first committed step. So the first thing that we can talk about is that idea of allosteric regulation, where you have something that's going to bind to that enzyme and either increase its activity or decrease its activity as a result of that binding. So there's a conformational change when that effector binds that's either going to make the active site more conducive to catalysis, so that would be an activator, or less conducive to catalysis, which would be an inhibitor. So we spent a lot of time talking about this sort of thing last semester. So effector binding influences enzyme activity. And again, we can have positive or negative effectors. So positive would then be activators, negative inhibitors. So we will see both. So that's our first mode. Another thing that we can do is we can covalently modify our enzymes. So that covalent modification can in effect serve as a switch, an on-off switch for our enzyme. Again, we spent a fair amount of time last semester talking about covalent modification as well. So adding or removing a group to the enzyme affects activity. So one really important one that we'll see a lot this semester is phosphorylation, dephosphorylation of the enzyme. So again, it kind of serves as an on-off switch. So very, very effective way of controlling the enzyme's activity. So kind of a new one, kind of goes in line with allosteric effectors, is something known as a substrate cycle. And so just to kind of describe what happens in a substrate cycle, I want to, before I talk about it, because I think it'll make more sense if I do it this way, is to actually show it to you. Come on, screen. There we go. So I have a few other figures that kind of go in advance of this. Again, this is really just the beginning. So this is those pathways on that one sheet of paper that were highlighted in dark, that were gray. So these are our initial catabolic pathways that we're going to talk about. So glycolysis being the first one, then we're going to feed into the citric acid cycle, and ultimately into electron transport and oxidative phosphorylation. In doing so, we're going to release a whole boatload of ATPs once we get down to oxidative phosphorylation. We'll do a little bit of ATP release in glycolysis and a little bit also in the citric acid cycle, but the bulk of the ATPs are going to come out down here. So those are our central catabolic pathways. This is figure 14-3 in your book, and it just kind of shows just a very general overview of what we're going to talk about. When we were talking about the catabolic pathways, we also talked about the fact that really what we're looking at is oxidation of carbon. So one of the things to make it easy to think about with respect to all these different oxidation states of carbon, which again I'm sure takes you fondly back to organic chemistry, calculating oxidation numbers, is really to think about where molecules are coming in as food and what our caloric output is for those foodstuffs. So we know that fats have more energy than do carbohydrates or proteins. Well, why is that? Does anybody know? Your clue is up here. What is the oxidation state of our carbons in fats versus proteins and carbohydrates? Are they more or less oxidized? Less. So our fats have more CH bonds than CO bonds. So they're coming in a less oxidized form. And so over the course of our metabolic catabolism, our processing of these foodstuffs, to get it from here, methane, up to CO2, which is at the top of our chart, our most oxidized form of carbon, there's a lot of opportunities to break bonds and form bonds to oxygen. And over that course, over that process, we can extract a heck of a lot of ATP. So there's more chemical energy, in other words, stored in these CH bonds than if you're starting more along the middle of the chart with amino acids and carbohydrates. So these are already partially oxidized. There's fewer opportunities for us to oxidize those carbons further, and as such, they're worth less on an energetic basis. So that's why when you look at your calories, why fats are worth 9 kcals per gram, and carbohydrates and proteins are only worth 4. They're more oxidized. So put it in that perspective, and I think it helps make it a lot more, have a lot more sense, and you can put it into perspective, because when we start looking at these pathways in detail, you'll see exactly what I mean. It's going to take a lot more processing if we start down here, and there's more opportunities to make ATPs as a result than if we're starting in the middle. Okay, so hopefully that will put it in a little bit of perspective when we're looking at it. Compartmentation, so this particular figure, it's 14-4, just kind of outlines within a cell. So we're not talking about different organs in the body, because we do also have compartmentation with respect to organs. But this at least tells you within the cell where certain pathways are going to dominate over others. So again, compartmentation is huge, not only on a organelle basis, but also within the organ systems of the body, so higher order compartmentation. Okay, so it's important to recognize that we're not doing opposing things in the same place, in the cell or in the body. And then in order to prevent our futile cycles, so this is just an unnumbered figure, it's, you know, alphabet soup, but what I want you to take away from this figure is that if in black we represent one pathway, so we're starting at 1 and we're ending at 2, we have to go through intermediate A. And that's going to require 2 different enzymes, both to make A from 1 and also to convert A into 2. But if we want to go in the opposite direction, and we want to go from 2 to 1, we can't go through A. We might go through some of the intermediates, but not all of them. But look at what we have. We have different intermediates and we have different enzymes to get there. So again, this is pretty cut and dried here. There are some things that are a little bit more gray in nature, but I think this, in a very simplistic fashion, gives you the idea that in order to do a conversion of glucose to pyruvate, it's going to require at least some enzymes that are different than taking pyruvate back up to glucose. Okay, so you have to have some things that are different. The pathways cannot be identical. Okay. So, substrate cycles. Substrate cycles basically are two different irreversible steps. So in one instance we have B going to C catalyzed by F, and in another instance we have C going to B catalyzed by R. And what really comes into play here is that what's going to drive that substrate cycle is some allosteric effector. And the allosteric effector is going to serve as an activator for one of these enzymes and an inhibitor for the other one. So if we have an activator of F, that will in turn be an inhibitor in R. So what that means with respect to a substrate cycle is only 1/2 of this is going to be operational at a time, because when you turn one side on, the other side is automatically turned off. Does that make sense? And so sometimes substrate cycles are really part of the pathway itself, and sometimes they are an accessory. So what we're going to see in glycolysis is we will see our first substrate cycle, and that's going to be regulation of phosphofructokinase 1, so the third step. And it's sort of a little side reaction process that's going on. And what it's doing is it's producing the major allosteric activator for phosphofructokinase 1. So again, we turn one side on, the other side is automatically inactivated. That is the basis of a substrate cycle. Both sides are not operational at the same time. What activates one inhibits the other. Okay, so we will see these quite a bit this semester. And I just wanted to spend a little time kind of looking at a simplified diagram because it's a little hard to explain in writing, right? So you have allosteric effectors that really are guiding this process with respect to the two irreversible enzymes. Okay. Any questions up to this point? Screen up. So substrate cycle characteristics: irreversible, opposing enzymes. Okay, so they basically have the same substrate product pair. They are catalyzed, irreversible opposing steps, catalyzed by different enzymes. Control is mediated through allosteric effectors. The allosteric activator of one enzyme is an allosteric inhibitor of the other. Okay, so in a nutshell, it's a little more complicated than this, but for now I think this will suffice. We'll talk in greater detail about it when we get to our first substrate cycle. Another means by which we can control metabolic flux is through genetic control. So turn genes on or off. This is a longer term control mechanism. So the first three are really the ones that are going to be the most prevalent, that we're going to talk about the most, because they can be activated very, very quickly. Genetic control to turn genes on and off is a little bit longer term sort of a commitment for the organism. But I want you to be aware that that's going on in the background as well. So it's not necessarily going to be something that we talk about, in other words, but it is a component of the regulation. So I want to just start, in the few remaining minutes that I have, a section that should be near and dear to your heart. And again, you know, we're just really reviewing some of the basic concepts that you've covered in other classes before, but putting it into context for metabolism at this point in time. And that is really to talk about, and I think this is such a weird term that I'm going to put it in quotes, high energy compounds. So other places you might hear them referred to as energy-rich compounds. I kind of like that terminology better, but your book uses high energy, so in order for there to not be a lot of confusion about it, we'll stick with that, but it's not my favorite terminology. So in a nutshell, these are molecules that release free energy when hydrolyzed. So of course, you know we're going to talk about ATP. That is our darling, right? But ATP is not the most high energy compound we're going to deal with. It's really kind of in the middle, which makes it perfect as energy currency because it can accept, in the form of ADP, a phosphate from a more energy-rich compound, and it can then donate from ATP a phosphate group to something lower in energy like glucose, so to make a less energy-rich molecule. So it kind of sits right in the middle. So we're going to talk first about ATP, and then we will, of course, talk about some of those other energy-rich molecules that you will see. And I will talk a lot about the structural characteristics that you need to look for. So if you weren't aware that something was energy-rich, and I just gave you a molecule and said, is this an energy-rich molecule? You should be able to tell me by the time we get to either the first quiz or first exam, just by looking at the structure of the molecule and the features of that molecule, whether or not you would consider it to be an energy-rich molecule or not. So ATP contains two energy-rich phosphoanhydride bonds. And again, when cleaved, free energy is released. So we can either do the following hydrolysis reaction. So ATP plus water gives us ADP plus PI, so inorganic phosphate. Delta G0 prime for this process, minus 30.5 kilojoules per mole. That's a pretty big negative number. Good, we like that. We can use that to help us. Or we can do, I think I can fit it on here, the following alternative. Instead of cleaving at that external phosphoanhydride, we can cleave the internal one first. So ATP plus water gives us AMP plus PPI, or pyrophosphate. When we do it in that way, we get a slightly different amount of free energy released, minus 45.6 kilojoules per mole. And I will tell you right now, I do not want you guys memorizing numbers. Magnitude and sign, much more important in this class. If we do that, I'm going to come up to the top because I have more room up here, so just kind of bear with me here. We can then do a second step because pyrophosphate still has that characteristic phosphoanhydride linkage, double, single, single, double. So I can still cleave that one and get out a little bit more. That one's minus 19.2 kilojoules per mole. Okay, so that one's a two-step transformation. And if you add up the numbers, if I go from ADP to AMP, it's about the same as ATP to ADP. So if you add up minus 61 kilojoules per mole for that route versus the second route, ATP to AMP, then PPI, you get a little bit more free energy out. We'll talk about why that is next time. Okay, so with that, I will see you guys on Friday.